computer. Alrighty. Thank you so much.
And then for that microphone, it's off right now, and it's just one button to turn it on, and the light will turn green.
Okay, cool. Thanks. Alrighty. I wanted to see, before I start, I actually want to test my microphone. Okay, cool. All right, nice. All right, hello everybody. Thank you for coming. I really appreciate it. My name is Matt Kane. I'm part of a new spatial company called Stellaromics, and here to give you a brief seminar and then some Q&A if you have questions after about a new company that was launched in 2022. I will be passing this plate around, but not yet. So I just want to have that ready. All right. So my name is Matt Kane. I'm one of two commercial people at the company. It's a pretty small company. It's about 50 people. And I cover the eastern half of the country. And so we're mostly trying to, get the word out, focused on early phase collaborations for new technologies. And so if you have appetite for that, this is definitely a really cool time to learn about this company because we're commercially launching now. I'll tell you a little bit more about our timeline, our technology, and then at the end, I'll tell you about some pilot program or other ways to collaborate with us as we kind of launch. Ready. So why did we do 3D? Well, biology is not inherently 2D, and pretty much every spatial biology method out there today is a two-dimensional technology. And they are great. They can answer a lot of questions, but they do not fit in certain applications. So if you have complex structures like vessels or lymph tubules or other three-dimensional tumors, teratomas, when you imagine that folding, tumor or folding vessel, if you slice through it in a 2D section on the left side there, you can see you're just kind of going to capture where it's passing through the plane of interrogation. But you'll really lose that 3D structure, and you don't actually know if all those dots on the left are connected or not. Same with things like cell-cell adjacency. If you're looking for different cell types and how they interact, they might be interacting with a cell just above or below your plane of interrogation. And if you can thicken up that area and capture that, you're going to get much more rich data sets to kind of understand the biology better. And then the third example we have here is just sparse objects. Sometimes if you have a tumor that's getting infiltrated by immune cells and you're looking at tumors that are cold and not infiltrated, well, if you don't actually look through the whole tumor, you don't know if you have some rare T cells that actually made it in there. So just by thickening up that sample type, you're increasing your data richness and you're increasing your ability to kind of catch those interactions. So this is what a typical 5-micron section looks like. So this would be any of those normal 2D technologies that are out there today. There's a lot of information you can get from this, but it is a pretty small area. And when you thicken this up to 100 microns, like we can do here at Stellaromics, now you're actually capturing, 10 layers of cells. So just in any of these, XY coordinates, you're going to about 20x the amount of cells that you capture. And so If you're looking for cell-cell interaction or rare cell interrogation, you're going to get a much better understanding of the biology in this 3D format. And this is what this looks like in a mouse brain. So this is actually a 50 micron slice of mouse brain. What we've done here is we've applied our panel of 250 genes to this tissue sample, and then we've used those 250 genes to cell type by different neurons and other cell types found in the brain. And you can see here, as we kind of zoom in, the RNA will kind of pop up. And so you actually have the X, Y, and Z coordinates for every single one of these RNA targets within the cell. So you can do things like nuclear or cytosolic genes. You can do cell typing. You can see, what genes are being expressed near other cells with those same genes expressed. So just a very rich data set. And lots of questions can be answered from these type of technologies here. If you're just thinking about data richness as well, anytime you're looking at a 2D technology, you might have, a certain area of your tumor that you just want to see, what's kind of happening. And so just having more cells, is valuable as well. So not only do you now have that 3D format, but you can see just the density of going from 5 to 100 significantly increases so that you just have more information about what's happening. I'm afraid to use my laser pointer because, it's not going to work anyways. Um, Also, the folks on Zoom can't see the laser pointer, so let me see if I can use my mouse.
Is this a typo of 100 micron?
100 microns is our thickness.
Yeah, typo. It says 5 versus 10.
Oh, sorry, that is a typo. You are correct. Yeah. Yeah, so for 5 versus 10, if you had an interesting structure here in this region, you may only have in 2D 20 cells in that area, but when you move up to 100, now all of a sudden you have 400 cells. So just the amount of data that you can generate to answer questions is a lot richer. And then this is showing 10 micron. Some of the 2D you can go up to 10 microns thick. So it's a little bit more information. Maybe instead of 20 cells, maybe now there's 40 cells here. But for the most part, it's very different in terms of the cell density. And when we actually calculated the cell numbers around this in mouse brain, we saw about 4 cells per square 100 microns, whereas when you go to the full thickness, it's about 65. So that's about 20 times the cell density. But not only density is important, so also things like cell nearest neighbor. So this was a study we did in skin cancer. And so we actually did cell adjacency analysis. So this heat map on the right here is showing basically nearest neighbor analysis, pretty standard 2D spatial technology. But if we shift this, I don't know if my video is going to play. Hold on. Let's see here. Okay. So when this shifts into 3D, you can see the heat map has completely changed. And that's because you're now capturing the layer of cells above and below. So having more data of which cell types are actually nearby physically in the plane above and below in the Z-stack is actually completely changing the way this heat map looks. And so if you were going to do this in 2D and draw conclusions from it, you might be missing something. So that's, definitely one advantage to 3D. Let's see here. Okay. Another example that I really like to talk about is actually in plaque. So there's been a lot of really cool studies done in 2D in plaque. You can definitely do distance analysis from a plaque and kind of see how the cells around there, maybe their glial cells are kind of interacting with the plaque. There's definitely a lot more biology to be discovered here. And if you're asking questions like how the spatial patterns shift or what are these molecular markers that are nearby plaque, this particular plaque, you don't know if it's the middle of the plaque. Is this the thickest portion of that plaque? Is this The bottom portion of that plaque, it looks like there's maybe a little bit of plaque over here. Is that the tip of the iceberg for an adjacent plaque, or is this just, an early developing plaque or something? So maybe these cells over here are actually nearby a big plaque and you just haven't gotten it in that 2D plane. So in 3D, you can clearly see that we're able to actually capture the entire plaque in 3D in those thick, either 50 or 100 micron sections. So we've stained for plaque here, and we're visualizing plaques of all different sizes. And then we can come in and layer all the different cell types that are near those plaques and just get a much better understanding of the cell biology that's happening. And what that looks like when you bring in the cell data is all of these different cell types and all the genes that they're expressing. So in this image here, the cyan is DAPI, and then the 4 different colors are all of the different genes that are expressed kind of outside of the nucleus near those plaques as well. And you can see we're still able to catch those different size plaques. There's some plaques that are large, some plaques that are small. And so if you're able to analyze that, you could think about, grouping cells by something very simple, like, if they're near very large, medium, or small plaques. And so what jumps out to me here is these microglial cells tend to be near the large plaques. So this is microglial here, but definitely a lot of, interesting information, definitely hypothesis driving questions could be answered from all this. So that was one good example, but before I continue on, I kind of want to back up and talk about how it's even possible to do this, because it was quite a bit of an effort to get here and is still quite a bit of an effort as we launch this technology. So if you're going to do 3D spatial, you really need 2 things. You need a way to see through the sample, and you need a way to image it with a piece of hardware. So there's a lab at Stanford University who's run by Carl Disseroth. He's the person that invented optogenetics and then clarity, which is clearing out, a whole mouse brain to look at, gene expression in the brain. And then his postdoc, Xiao Wang, is now at the Broad Institute, but she, in his lab, invented this star map chemistry. And I'm going to go through the whole chemistry in a moment. But this chemistry relies on the ability to have this specific sequencing called CDAL sequencing, and it also requires a confocal microscope, and it also requires clearing the tissue. So she kind of built on the expertise of Carl to kind of do the sequencing in the cleared tissue. And you could theoretically do StarMap in any confocal instrument, but the decoding process and the clearing optimization is really, really challenging. And so this paper came out in 2018, but no one was really using this technology. And so in 2022, our company was founded to commercialize it and make it a lot easier for everybody. And so 2025 is a big year. We just did our Series B round of funding, and we are shipping out our first few beta instruments now. So we have one going to Glasgow in Scotland, and then we'll have one going to California in about 3 weeks, and then one going to Emory University in about 6 weeks. And then next year, we're going to be launching the commercial version of the platform after we finish with that beta phase. So what does that instrument look like? So this is the hardware. It's basically a thermally controlled box with multiple cameras, confocal microscope in there, a lot of Z scanning ability that's we've developed intellectual property around. And then we actually put our own NVIDIA GPUs behind that panel on the bottom. So if you were to take off this bottom panel, the bottom half of the instrument is basically just stacks of NVIDIA GPUs. And the reason we do that is because when you're doing thick tissue, if you're getting 20x the amount of cells, you're getting 20x the amount of data. And so we will live process 800 terabytes of data from just 4 samples simultaneously on the instrument. And then you actually only export a few terabytes of decoded data. So all of the primary analysis happens on board. There's no cloud processing. There's no cloud subscription. It all happens within this piece of hardware here. And then the top half is basically a thermally controlled box with an airtable. So whenever you're doing Z stacking, you need to make sure that the instrument's very stable, which is why we've built an airtable in there. And then we've sealed it up completely so we can control the temperature so that there's no expansion of the metals or plastics that we're going to be using in there. And then we have multiple lasers, multiple cameras, multiple optics to basically do all this confocal imaging. Um, and so we've kind of taken the hard part of building something like this. And I also don't think that this was possible before recent advancements in machine learning and chip hardware. So now is kind of the time to be able to do 3D for the first time ever, which is pretty exciting. This is me with the instrument. So it's about the size of a NovaSeq. It's about, 5 feet tall or so, and it sits on the floor. And then one quick pitch real quick that I'll just go through. We are doing the commercial release next year. So we're going to do a 3D Leaders program. So this will be priority access to the first 15 instruments or so. You'll get 2025 pricing, and then you'll get early access to the first shipments, which will probably happen in early next year, maybe Q1, Q2. And then you also get the ability to work with us on certain tissue types. So we're optimizing a handful of tissue types I'll go over in a moment. But if you have a certain tissue type that you really need to see in 3D and you join our 3D Leaders program and you're part of those 15, then that tissue type gets kind of prioritized by R&D, which is kind of nice. All right, so now I'm going to talk about the workflow. So right now we're doing fresh frozen blocks, usually in OCT. This is the plate that they'll go in. I'll pass this guy around and just check it out. So anything that you can fit in that plate, you can analyze on this instrument. So typically you'll take your sample, whether it's a mouse brain or tumor tissue, slice a either 20, 50, or 100 micron slice of that, plop that into the plate. And then there's about a 3 or 4 day sample prep protocol here. So the first step is we're going to generate these amplicons. These amplicons bind to your mRNA. We have 2 probes here. We have this leading probe and then this padlock probe. Only when these 2 probes bind to the mRNA does this nick ligate. And once that nick ligates, then we can do rolling circle amplification to generate these amplicons. So we call this our snail probe because it kind of turns into a little bit of a snail. And this little gray region here has a few nucleotides that we're going to sequence in 3D. It's like a barcode for each gene. This amplicon is very stable. It's actually DNA. And it also has small acridite groups in the backbone of the DNA. And that actually is what gets embedded in the hydrogel. So when we ship you the sample prep kit in that plate, it'll actually have hydrogel on the bottom, and then we will grow the hydrogel up through your tissue. Once the hydrogel is, or once the tissue is embedded in the hydrogel, we actually clear the tissue out, and then that makes it optically clear so we can stack all the way through the image. This is what the sample prep process looks like. So a lot of folks are used to putting, slides in a microscope. We found the well plate makes it a lot easier to control the chemistry. And so we've come up with this method to just use these little transfer rings to basically pick up and plop the sample in the well like that. It stays nice and flat, and then it kind of melts onto the hydrogel in the back there. Very easy to do. If you ever worked with a cryostat, it's pretty easy to slice 50, 100 micron sections. And then it gets sequenced. So the Pyxa is a 3D sequencer. So we'll cycle multiple fluorophore cycles to get the individual nucleotides in that backbone. And then we'll do all of the primary imaging processing on the instrument. And then the export is actually a very typical .csv file of cell by gene matrix. So if you've done single cell work in the past with Syrah or SquidPy from spatial methods or single cell data like Chromium Rhapsody, you'll be very familiar with the raw data that comes out of this instrument. And so that can be processed in third-party tools, or we have our own visualization software that I'll show in a moment. But before I do that, I just wanted to go one level deeper on the chemistry because I find this stuff really interesting, and people usually like to see exactly what's happening. So the way that we're sequencing that barcode is actually with a method called CDAL sequencing. And if you want to really understand this, you can read that 2018 STAR Map publication. But basically what's happening is we have four different fluorophores and then ATGC. And so the reason we're 250 plex right now is because we're reading four nucleotides. We plan to go higher plex next year and in the years to come. So when we launch the instrument, it'll be 250, but the hardware will be able to do higher plex over the long run as we basically increase more cycles. So to start, we'll have those four colors, do ATGC across four nucleotides, and that's how we get to that 250 plex. There's a lot of advantages to this dual probe design. It's very specific. So because you have both the leading probe and that padlock probe, we know that we're binding the accurate target. And then this can be a good backbone for future assays like protein, siRNA, guide RNA, other applications than just mRNA, which is pretty cool. And then we have our Pyxa Studio software. So the Pyxa Studio software we kind of built to visualize 3D. There isn't really a great 3D open source platform out there. So we built one for the qualitative and a little bit of quantitative visualization. So in Pyxa Studio, you can do heat maps, U maps, clustering, visualization, and then you can also offboard your raw data to the cloud. This is just a screenshot of Pyxa Studio. So you can see on the top right, those are all different cell types, and you can see all the colors. You can kind of pan through to look at different layers of your tissue. And then the bottom right is all the different genes. So you can turn genes on and off. You can really do a lot of qualitative analysis and visualization. I like to show this because it's just kind of cool to think about. So this is the 49 people that work at this company. This is me here. So we had a company retreat 2 weeks ago to kind of celebrate launching our beta unit. So we just shipped that to Glasgow last week. And so that's really the first instrument to go, which is a very exciting time for us. And the company's been building over the last few years, but we're really at this exciting launch point now. And this is our Boston team. So I'll talk a little bit about some kind of pilot options with them in a moment. And then this is our team that actually helped build and ship out our first beta unit. So you can see in that crate behind their head there, that's actually the first instrument shipping out to Glasgow. So if you'd like to work with these wonderful people, we have a couple scientists in Boston that can do pilot projects. We do have this pilot program that we call our Technology Access Services. So right now we're doing mostly mouse brain. We have a couple other options for some tissue types. And so it works by sending us a block of brain, and then we'll do all the sectioning and put it in that plate. We'll do some primary analysis for you. If you want to add in some custom genes, you can do that. We can do things like the Alzheimer's stain or tau tangle stain. We can do cell by gene matrix delivery or help you with some heat map analysis or things like that. But the reason we really do this is just to kind of have people test the technology. Most people might not have heard of Stellaromics. They might not have worked with 3D data in the past. So we're just trying to kind of get the word out and generate some of these amazing 3D projects that can really only be answered in 3D. So what that looks like is basically an initial call and a quote. There's a project kickoff meeting, and then you ship the blocks. Our services team processes the samples. It's about 2 or 3 weeks to run, and then 2 or 3 weeks of data analysis. So the total time is usually about 6 to 8 weeks. And definitely chat with me if you're interested in this at all. This technology has been demonstrated in a few different tissue types, and some of them have some really interesting 3D applications. So these are kind of our early tissue types we started to work on. So another one is human brain, kidney, liver, retina, and tumor. So retina especially has a lot of really interesting 3D kind of architecture to the way that the cells are layered, but it's really hard to kind of clear all the pigment out. So we've been working on this one for quite a while and plan to launch it commercially soon, which is exciting. And then, genes or tissues like kidney and tumor definitely have a lot of 3D architecture. So if you'd like to kind of work with that, we would definitely like to chat with you. And then just in general, I mean, where we see this having the most application is in areas like neuroscience, oncology, if you're looking at rare cell populations, tumor infiltrating lymphocytes, gene therapy. We're working with a lot of pharma companies around things like guide RNA, siRNA, et cetera. So if you have a oligonucleotide therapeutic and you want to know the cell type that oligonucleotide has gotten into, um, you could definitely do that with this technology, excuse me. And then also looking at different immune cell types and their spatial organization. All right. We also are in R&D working on a few more projects that are on our roadmap next year. Um, so a couple of them are things like guide RNA for CRISPR work, ribomap, which is actually the ability to see which genes are actively being translated. So you could see, is that gene connected to the ribosome, which is really cool. And then also doing things like rare cell, rare event detection. We're also starting to work on organoids as well. So if you're doing any kind of pharmacology screening, um, having the plate is nice because we could theoretically do live cell imaging. Um, we are also working on viral labeling like AAV, rabies virus. If you have a viral vector and you want to see how that's affecting your cells or use it to label your cells, we've definitely done that. We also have technology and development around protein. So we have a proprietary way to kind of get the protein expression targeted in the cleared hydrogel. And then I also just wanted to talk briefly about the ribosome. So I mentioned this 2 slides ago, but the way RiboMap works is we basically take our normal padlock and leading probe, but then we add in a third nucleotide, and the third nucleotide or oligonucleotide, excuse me, is a sequence that is targeting specifically the ribosome. So this complex will only assemble when that mRNA is actually in a ribosome. And so what that tells you is if those amplicons are lighting up in your tissue, then you know that that's being actively translated. So if you're working with some kind of cancer or treatment sample and you want to see like what are the genes that are actually turning on as I'm kind of inducing a change in my samples, this is the technology that can definitely do that for you. I like to talk about other kind of trickier sample types as well. So this is one proof of concept we did in colorectal cancer. It was only on a small panel, but we've definitely been able to show that even in these really weird tissue types, we're able to kind of clear it and get good data from different genes and cell cycle genes. You can see that in the top left in the red and the blue there. And then we're also working on other kind of really interesting tissue types, like this is liver in 3D. So this is still very much future directions in R&D, but we're actually working on getting the morphology of cells. So if you actually have questions about the physical shape of your cells, and staining for that in 3D can actually tell you, the morpholome of your cell types, which is very interesting. It's a little choppy, but you can kind of get the gist here. A lot of times when you have a tumor being treated, the cell phenotype can change and just look different. And so you can't really do that in 2D because you're just kind of collecting data. But in 3D, if you can actually confocal and see the shape of the cell, you can get a lot of information as well. So this is another future direction. My last 2 slides, one I just really like to mention is our leadership team here. One of the reasons I really joined the company is because we have a lot of kind of experts in the industry. So our CTO is Mark Pratt, and he was one of the Solexa employees, which is part of that early Illumina acquisition that started basically Illumina. And then Todd, our CEO, was, I think, employee number 8 or 10 at Illumina. And so we have a lot of really strong leadership that have basically been able to build companies like Illumina from the ground up, and now they're here at Stellaromics, which is awesome. And then our chief scientific officer, Ye Fu, he was in one of the Harvard spatial biology labs that actually developed the MERScope. So he brings a lot of spatial expertise as well, and now kind of helping us launch 3D for the first time. So really great folks to work with. And the cool thing about a small company is we I work with these people every week. And if you do pilot projects with us, you'll definitely get to meet and chat with them, which is awesome. Just to reiterate on our timeline, so this year we're launching our 3 beta instruments. I can definitely talk to you more about that after. And then next year we have the early access for 15. And then late 2026 we'll be doing standard commercial. So I pull this slide up last just to kind of reiterate how exciting it is to work with a company as they're launching. I think if you can intuitively understand that 3D's going to answer some biological questions in your tissue type and you want to be involved with a company that's doing it for the first time, you will be among the first people to generate 3D data. And with that, I'll say thank you, and then I can take any questions. I might pause the recording. But yeah. Any questions?